In this lecture, I am going to discuss about the replication of DNA. So, replication of the DNA is the synthesis of new DNA strand by using the parental strand. As in this diagram, you can see that this is the old DNA strand. First, it gets unwind, and after that, on that strand, new DNA strand is synthesized. So, these are the various steps of DNA replication. Number 1 is initiation, number 2 elongation, number 3 termination, number 4 proofreading and correction. This is a bacterial DNA, it is circular, and this is eukaryotic DNA, it is a linear. So DNA replication is start at particular location. That location is called as origin of replication. In a bacteria, there is a single origin of replication is present from where the DNA start to unwind, while the eukaryotic DNA consists of the several origin of replication where the DNA unwinding is start. In the first step of replication, that is initiation, the unwinding of DNA double helix takes place, and the unwinding of this DNA double helix is performed by the enzyme called as a helicase. This is a helicase enzyme which separates the DNA strand into the two template strands. And how this helicase separates these two DNA strands? By breaking the hydrogen bonds present in between the nitrogen bases. So in this way, this DNA helicase is responsible for the formation of the replication fork. Now, ahead of the replication fork, there is so much supercoiling takes place, and this supercoiling is loosed by the enzyme called as topoisomerase enzyme. This is a single-stranded binding protein which help to keep the parental strand apart. Next function is performed by the enzyme RNA primase. That primase enzyme adds a short sequence of the RNA to the template strand. This short sequence of RNA primer which allow the DNA polymerase enzyme, which is the main enzyme of the DNA replication, to bind to the strand to start the replication. After the addition of the primer by the primase, the next enzyme comes in a role, that is DNA polymerase 3. DNA polymerase 3 add a nucleotide to each template strand. Okay, this red color shows that the nucleotide added by the DNA polymerase 3. So this DNA polymerase 3 add a nucleotide in a 5' to the 3' direction. So this is all about the initiation process in which this enzyme play a important role. Now we will discuss about the elongation. Since the strands are anti-parallel, means two strands have their 5' end and 3' end in opposite direction, but the replication only occur in a 5' to the 3' direction. As a result, one of the strand will replicate it in the same direction of the replication fork, and the another strand will replicate in the opposite direction of the replication fork. So the strand which is synthesized in a continuous manner, the strand is called as leading strand, and the strand which is synthesized in a discontinuous manner, that strand is called as lagging strand, and each fragment of the lagging strand is called as Okazaki fragment. After that, DNA polymerase 1 comes and remove the primer and replace this primer to the DNA nucleotide. And the last enzyme, that is enzyme DNA ligase enzyme, that joins the Okazaki fragments together to form a continuous strand. Last is the termination process, that is to stop the progress of the replication. As the bacteria have a circular chromosome, therefore termination of replication is occur when two replication forks are made to each other. While in a eukaryote, DNA replication initiates at the multiple points in the chromosome. So the replication fork meet and terminate at many points in the chromosome. So proofreading and correction is performed by the DNA polymerase, which make a very few errors, and most of those that are made are quickly corrected by the DNA polymerase. If the newly added nucleotide is not complementary to the one on the template strand. This enzyme remove the nucleotide and replace it with the correct one. DNA is copied with the less than one mistake in a billion nucleotide. So this is all about the replication of DNA. Thank you.